Hello. So let's get started. So hello everyone. So I'm Fred Saudou. It is a great privilege today and an honor to introduce Professor Jean-Louis Mandel for this keynote session. Jean-Louis Mandel is the scientist that had to be invited today because he's among the scientists that discovered and characterized unstable genetic mutations. And this includes Huntington disease. This is thus a tremendous opportunity to highlight the important contribution of Jean-Louis to the field. Its international recognition is a major and it go beyond international, and Jean-Louis is a major figure of medical research in Strasbourg, and this is particularly important in the context of this European EHD network meeting this year in Strasbourg with the 20 years of EHDN meetings. Finally, as a former researcher in his lab that I joined just after the identification of the Huntington gene, Jean-Louis particularly influenced my career. So briefly, Jean-Louis is a doctor of medicine and doctor of science from the University of Strasbourg. He completed his thesis under the supervision of Pierre Chambon during the time during which he published seminal articles about the presence of introns in the ovalbumin gene at a time RNA splicing was still overlooked. Jean-Louis Mandel became in 1978 Associate Professor of Biochemistry at the University of Strasbourg, Faculty of Medicine, and then became Professor of Medical Genetics until 2003, which is the time he was appointed as Professor at the Collège de France at the prestigious chair of Human Genetics. Since 1980, Jean-Louis has led Human Genetic Research Team first at the Laboratoire de Génétique Moléculaire des Eucaryotes that was directed by Pierre Chambon, that then became the Institut de Génétique and Biologie Moléculaire and Cellulaire, the IGBMC, in 1994. In parallel to this very intense basic research activity, Jean-Louis founded in 1985 the Laboratory of Molecular Diagnostic for Genetic Disease at the Strasbourg University Hospital in 1992. And this became an essential instrument to diagnose patients, but also to identify and characterize genetic mutations. Jean-Louis was appointed director of the IGBMC from 2002 to 2007. He has also been acting as director of the mouse clinic in Strasbourg, that is a major instrument for developing mouse models for many diseases. Professor Mandel had a major contribution in the field of human genetics by discovering in 1991 an unusual mutation in a gene of the X chromosome that causes Fragile X syndrome, or XFRA. XFRA is an inherited form of intellectual disability. And at the time, Jean-Louis and his colleague, the late Isabelle Oberle, showed that the mutation was a string of triple-letter repeats that disrupted the particular gene, the FMR1 gene. So today, unstable repeat expansions are recognized as a common pathological mechanism responsible for more than 50 genetic disorders, and among them is Huntington disease. I will not go into the details and the importance of the discovery, as Jean-Louis will tell you more in a few minutes. In addition to major contribution to understanding XFRA, the team of Jean-Louis Mandel also identified the cause of many other genetic syndromes, and that includes just Friedreich ataxia and several spinocerebellar ataxia genes that are also characterized by unstable mutation. Jean-Louis Mandel is a member of the French Academy of Science and of the National Academy of Medicine. He's Knight of the French Legion of Honor, and along his career, Jean-Louis received many prizes, among them the 2022 Kavli Prize in Neuroscience that was jointly attributed to Arior, Chris Walsh, and Uda Zogby. He received also the Louis Jantet Prize of Medicine from the Swiss Academy of Science, the Grand Prix de la Fondation pour la Recherche Médicale, and also the Richard Lounsbury Prize from the French and US Academy of Science. I would not close this introduction without saying that besides this exceptional scientific career, Jean-Louis definitely forced our admiration because Jean-Louis is also an expert in art. He can make you visit the Museum of Strasbourg, and he's also an accomplished violinist, and at the time he had to choose between a career in the violin or in medical research, and I think we are very lucky that he chose medicine and genetics. Thank you, Jean-Loup. This is a great pleasure to have you, and the stage is yours.
Okay, I don't know if I can survive this too laudatory presentation, but thank you, Fred, and thank you for inviting me, although when I started to prepare it, I thought, you know, how I'm going to do it in 30 minutes, talking about the history of unstable repeats with now about 50 or 60 different disease and genes. But I should also tell something that you certainly remember. We started to work on Fragile X, indeed, in 1983, as I'll tell it. And we discovered the gene and the mutation in 91. And we had also a DNA diagnostic lab. And even before the Huntington gene had been really identified in 93, in this diagnostic lab, as we knew where was, in which region of the genome was the Huntington disease, we had started to do linkage diagnostic for genetic counseling in families who were asking to know whether they had inherited the wrong part of the chromosome or not. And so I knew about Huntington. I was very interested. And when Canadian postdoc who came to the lab, he was supposed to do a mouse model for Fragile X, which involved cloning in bacteria this extended CGG repeat. And he couldn't do it. It was failing. And he had a control, and the control was a CAG repeat. And the CAG repeat was easy to clone. And I think it's Fred who said, so why wouldn't, when the Huntington gene was finally identified and found to be a CAG repeat, he said, so if it were, the CAG repeat works, why wouldn't you do a Huntington model? And this is what we did, and he contributed to the beginning of the story on CAG, was actually the cloning of SCA2 and SCA7. He was part of the team. So thank you for this early contribution, which was very important. Now, this is our beautiful institute, IGBMC, on the outskirts of Strasbourg. And what do? I do this, I think, yes. And there's this international meeting, European meeting here in Strasbourg, and I feel like we say for the Tour de France, I am le local de l'étape, le régional de l'étape. So this, you know, the guy from the region who will try to do, when the Tour pass in Strasbourg, there should be some champion in Strasbourg that try to do as well as he can. So I am the local de l'étape. But I must say that since 1991, and I'm very proud of this because this was done mainly by people who started working with me but became independent investigators, or even better, not better, but by people who came to Strasbourg not having worked with me before and established very good, superb groups working on some aspect of unstable repeats. So we have been working over this now more than 30 years on Fragile X, Friedreich ataxia, SCA2, SCA7, where we actually indeed identified the mutation. And there was also in Strasbourg major work on mechanism, cell and mouse models for this disease, other polyglutamine expansion disorder, Friedreich ataxia, myotonic dystrophy, ALS. There is now very active groups there. And the newly identified, or rather newly, polyglycine disorders. And we have been doing also quite a bit of diagnostic and genetic counseling applications. And maybe another story that Fred Saudou knows is on the paper is showing how serendipity can help researchers, because there was a team in IGRMC working on transcription, and especially on a transcription factor that was named TBP, Tata binding protein. And once Yvon and me, we heard a talk by a member of the team who was trying to get very specific antibodies for this protein. And then he said as an apart thing, yeah, I have also an antibody against, there's a poly-Q repeat at the end of this protein, and it's of no use because it's present in other proteins, it's not specific, so, you know, we discard this antibody. And we came to tell him, you know, that could be very interesting because actually the Huntington protein at this poly-Q, so maybe it could be a useful antibody. And it ended to be a miraculous antibody called the 1C2, used in many, many labs, because it was actually detecting in the cells from patients only the pathogenic form, not the normal form of Huntington. It was detecting only the expanded. And then we could show with Alexis Brice and Alexandra Dur, who had this bank of people with various movement disorder, that it was true also for several ataxic patients where it would detect only expanded but not normal. And this way, actually, we discovered that several ataxia were actually polyglutamine disease. And this was also a major paper here. And this was then distributed to many, many labs and was useful to actually detect these aggregates. Eric, I don't know whether you're still using the 1C2 monoclonal antibody. So this is 20 years later. Okay, so let's come. There are other things to do around Strasbourg, especially when you're interested in unstable repeats. And another, with Christopher Pearson and Laura Ranum, we organized an unstable repeat meeting in 2012. And you see here many luminaries, the names are there, working on, or who did great work on unstable repeats. And I would point out that actually I went back to a very good book to prepare this talk, and which is called Tomorrow Belongs to Me. And this is by an artist from Scotland who actually comes from a family with myotonic dystrophy. And all this thing about what will happen, you know, what is the tomorrow, and whether she has or not the gene, whether it will start earlier because of anticipation. She was very interested, and she then made a project with a very good geneticist colleague, Darren Monkton, and they went all around the world to interview key people who played a big role in identifying expansion disorder in Huntington, in myotonic dystrophy, and in Fragile X. And you learn very interesting things from this book. And I decided to actually start, I'm only starting, with the concept of anticipation, because there is a very interesting story for scientists, for young scientists, I think, and also for, I think, for families, that sometimes the knowledge is in the families, but for some reasons there may be difficulty for the scientists to accept this knowledge and find reasons that maybe this knowledge is no good, but it is. And what's said of this, Kurt Fischbeck, who actually identified one of the diseases, polyglutamine, the first actually polyglutamine expansion disease to be discovered, and who is a clinical neurologist also, so he was seeing families. And what he says, families would see it. They had come to the doctors and say, it is clearly getting worse in our family, and it would be hard to say to those family, you know, you're just a statistical aberration, because this was the interpretation at the time of this phenomenon of anticipation. So I will tell you. And of course, I want to say how, of course, Nancy Wexler, that I met several times as we were working also on Huntington, played this major role in actually getting to the gene and to support research on Huntington, a very important role. She is interviewed indeed at length in this book. And also to two great, well-known scientists, Sir Peter Harper, who did major work on both myotonic dystrophy and Huntington, and Steve Warren, who did major work on Fragile X, and my young colleague, Isabelle Oberle, who didn't have a chance to become famous because she died at the age of 35, the year she played the major role in identifying the mutation in Fragile X. That was in '91, and she died the same year of breast cancer. So these are the topics that I would like to address. And Fred, tell me, have I already finished? Okay. It will be the fight between him and I. I'll try to speak longer. He will try to prevent me from doing so. But if I can't do all, it will be available so you can have. Now, I'll start on anticipation because, as I told you, I think it's an interesting lesson. My slides on this are unreadable because they are not there for you to read them, but for me maybe to remember what I want to say. If I can see, wow. Okay. I will talk about anticipation and other bizarre familial transmission patterns, especially in myotonic dystrophy, where anticipation was defined, and also in Huntington, where there is a special way, and Fragile X, where there is another bizarre transmission pattern. So anticipation is a concept that started to be used in the early 20th century, and especially in the context of mental disorder, but not only, also even of tuberculosis, diabetes, etc., that maybe for some disease it would starting to get worse or start earlier and be more severe in successive generation of families. And this was a concept that has strong eugenic connotation, you know, because for some people at the time, there was this risk that people with bad genes was just a name, you didn't really know what it was, but would lead the population to get worse and worse because they would have, you know, in successive generation, more affected children, etc. And maybe you would like to prevent this. And actually, there was one disease, myotonic dystrophy, where in 1918, a German guy published that indeed in this gene you could see clearly anticipation, because in successive generation the disease would start earlier, age of onset. It's a disease with very varying age of onset, like Huntington. Even worse, because there is an important congenital form where babies are affected very severely at birth. So even worse than Huntington for that aspect. And so he said, yeah, there is anticipation in myotonic dystrophy, and it became you know, something part of the knowledge. Until in 1948, a prestigious British geneticist, very respected, Leonard Penrose, and who is also statistically minded, made calculation on his hypothesis that actually anticipation was maybe just a bias in looking at families. Because when you look at families, you start with somebody who has a very severe thing that can be easily diagnosed. I heard Alexandra saying that some people with short expanded repeats, Huntington, may not get diagnosed because they have no chorea. They may have some kind of cognitive impact, but since they don't have chorea, they are not diagnosed with this. While a guy who has a really important movement disorder, you diagnose it. So I was saying, you tend to see the more affected, and then you look in the family, and of course you see maybe somebody less affected, because if he was as severely affected, maybe he would not have kids. And so he made this calculation, no epidemiological data. There was no data that he was analyzing. He was doing statistic on a hypothesis. And his hypothesis was that anticipation is an artifact of the way you ascertain patients and families. And because it was prestigious, this became dogma. Anticipation is a statistical bias. Doesn't exist. There is no biological basis. In a very good, prestigious textbook on human genetics in 1986, it was said clearly, anticipation, like in myotonic, was proposed for myotonic dystrophy, is a statistical bias that has no biological meaning. And then no, sorry, the wrong. When there was a rather young neurologist, clinical neurologist in Nijmegen, who started for a thesis, PhD thesis, to work on this anticipation problem because he had seen many families with myotonic dystrophy. And he thought, it's curious, you know, I see the anticipation. Why should it be a statistical bias? Or is it really true? And when he started to work on this, people said, but why are you working on something that is solved? Penrose said in 48 that it is, you know, an artifact, so why are you working on an artifact? And, but he continued, started in 74, as he was also a clinician, took time, and also to follow families. He followed the whole families, you know, extended families, and started to see whether the arguments from Penrose were adapted to what he was saying, and it was not. And so he did his thesis, and he had difficulties to find people who would support the thesis. As I said, many said, no, I don't want, because they all said, well, this can't be true, because it's proven already that anticipation does not exist. Some wrote to me saying, thanks for the thesis, but we don't believe it's right. And there was one, he asked for being the jury, who even didn't reply. And then somebody told him that he had said, you know, I'm not going to spend my time reading this bullshit, because this is bullshit. Okay? But finally, managed to get his thesis, wanted to publish it, had difficulties to get it published, and with the help of actually Peter Harper, who actually started to believe, even though he was a believer first, that anticipation is an artifact. He was convinced, and finally it was published in 1989. So Christian Haveler is a great guy because he really fought, and this is something that I would say, especially to young people. There is a tendency you cite things that you have not read. If it's really important, you know, because it's a title, maybe it's in the abstract, if this is really important for the continuation of your work or for your thinking, maybe you should read and see, is the paper as strong, are the conclusions as good as you think? So, this is anticipation. Now, in HD, is there anticipation? Actually, there was, I found that before the gene was found and with a CAG repeat expansion, there had been a few papers looking for anticipation in Huntington families. And actually, the curious thing, it's why I underline in core anticipation, that each time they speak about anticipation, is there anticipation? Or maybe there is, they put it in quotes as if it was a dirty word for a scientific paper, you know, anticipation. You know, we know it's an artifact, but, you know, let's still look. And actually, there was one thing where really it looked like anticipation. It was that the largest expansion with the juvenile cases, they always come from a father, and of course a father who didn't have this juvenile. So there was a lot, in this case, 20-year or more anticipation. So the conclusion was there this kind of anticipation, quotes, but only in some of the paternal transmission, not in the maternal. So this was something bizarre, not explained. People thought about imprinting, about methylation of DNA, maybe interaction with another gene, but I have to finish? Almost. Okay. So I see that indeed, so maybe to cut a very long story, too long story, but I like to tell stories. Maybe this is. So I will maybe say, just show you one slide. So this was the slide that made us famous in 91 with the expansion and an imprinting epigenetic modification also in Fragile X. This was very useful for diagnosis, for understanding the disease, etc. And we continued to work on this until now, on the mechanisms. But I want to tell you that after Fragile X, there was a polyglutamine expansion in the androgen receptor gene for this spinobulbar muscular atrophy, which is mainly a muscle disease, if you want, although it comes from motor neuron affection. And there, it was Kurt Fischbeck that found it with Al Laspada, who was his student. And when he sent it to Nature, it was actually rejected because, okay, what is this expansion? It was not unstable, so it didn't look funny or interesting. So it was rejected. But when the Nature editor saw the Fragile X story, they said, this looks a little bit, this SBMA thing looks a little bit. So they called back Fischbeck to tell him that after all, we're publishing. He actually had said it only 2 months before the Fragile X story was published, so it was not it didn't lose so much time, but this is an interesting story also that sometimes editors of big journals can miss important things. Then there was a myotonic dystrophy, and actually in this book, Keith Johnson tells that he had the idea that maybe myotonic dystrophy could be a repeat by hearing me at Cold Spring Harbor talking about Fragile X. And then, and Nancy Wexler has a similar story. She thought about repeats when she heard about Fragile X. And this was then Huntington, and the same year, SCA1 with Harry Orr and Nuda Zogby. You will hear Harry Orr at a keynote lecture. And then I skip all this to get there. In the first about 10 years after discovery of Fragile X in 91, there was about 15 repeat expansion disease that were identified. And then it started to slow down in 2000. And so many people, including me, thought, okay, all that could be found is now found. There will be no more or maybe extremely rare disease with expansion, but we know about this. There's about 15. And then a bomb exploded in 2011 with the identification of a repeat, a GC-reach repeat, very difficult to work on, in the non-coding part of a gene of unknown function, C9orf, called for, we don't know the function, for a disease that is the object of much attention, which is amyotrophic lateral sclerosis, or Charcot disease, or Lou Gehrig's disease, and it was found that actually not only was implicated the expansion in this gene could cause familial form of ALS or even sporadic forms, but also of dementia, frontotemporal dementia, and that these two things were two aspects of the same disease. That was very important. And this, then people started to see maybe we can find other, and especially because of the progress of technology, doing whole genome sequencing, doing long read sequencing, having bioinformatic tools to detect these expansions that are very difficult to detect bioinformatically. You need special tools. In the last 15 years, or 10 years, sorry, there has been more than 20 genes found, and maybe I'll end just, this is a list, and there are different repeats, different place in gene where they are, but they are now, it's not only a rare thing. Two of the most important late onset form of ataxia are actually due to expanded repeat that have been discovered in the last 2 or 3 years. CANVAS and FGF14. So these are very important discoveries for people with this late onset disease where one maybe didn't think it was genetic because it was late onset. And another astonishing thing is the FAME story. The FAME story is now there are 8 genes that cause this familial myoclonic epilepsy and movement disorder again. And what was astonishing, that is this, actually 7 have been discovered. There is 1 not yet, but 7 have been discovered. It's always the same pentanucleotide repeat. A complex repeat, it's a very complex story, but always the same in these 7 different forms. Same clinical expression. Yeah, I've finished. But the most astonishing, it's in different genes that have nothing to do one with the other. So it's not the gene where it is that is important. It's a mutation that tells whatever probably the gene it is. So I think this requires explanation. Many things requires explanation, and this is why what I thought would be a 30-minute ended to be 35 or 40, and I stop.
Thank you very much.
When you invited me.
Thank you very much, Jean-Louis, for this fantastic talk. And before we go to the break session, there is an announcement from Cristina Sampaio. And Cristina, welcome.
I am sad to stand here with sad news, and particularly after a wonderful talk, but I think it's important that this community learn about the passing of Professor Fernando Lopera last Monday. Professor Lopera worked all his life in Colombia, in Medellin, and he made impressive contributions to the field of neurodegenerative diseases, particularly for Alzheimer's disease, but lately he was working also in Huntington's disease. I know that many of you might know his work, but his work is worldwide renowned, but it's particularly important because he did it in Colombia, a country with small resources and a country that was in a civil war when he started to do all these painstaking research in families with family Alzheimer's disease. So all this very detailed work led to the discovery of one of the most important mutations in Alzheimer's called the PISA. Is seen in this picture, seeing the patients. He was known by his empathic touch, by his ability to reach to the people, by his ability to build collaborations, and he created these impressive genealogic charters of these large families. And so by his work and by what he was able to build in Colombia and particularly in Medellin, but the big team he created around him, I'm sure his legacy will survive him for many, many years and decades. I'm sure what he contributed is part of the global science. So I met him several times. I don't know him very well personally. I'm sure colleagues from Colombia would be better fitted to stand here to speak about Fernando, but I recognize that he was one of the big men of the science, and I know that particularly in Catholic tradition, when people passed, we are sad and sorrow, we should be, but I would ask you to applaud his life, to applaud his contributions to science. So please join me in commemorating the life of Fernando Lopera. Thank you.